Well, hey, AP, going to do lecture 2 now in photosynthesis. I'm going to do this on notability because to see if it's a little bit more smooth, but also to be able to draw and color one thing. Yes, we are going to be coloring, so you will need some color pencils for this lecture. And if not, there is this picture online that I'll show you towards the end of this lesson. Okay, so here's lesson number 2. Here's our objectives. We want to describe the relationship between wavelength and energy in the electromagnetic spectrum. Okay, we want to explain the role of photosynthetic pigments in obviously harnessing solar energy, and then we definitely need to examine how ATP and NADPH, this is going to be a big molecule today, how they are produced from redox reactions and membrane gradients. Okay, so light is a form of electromagnetic radiation. It's propagated as a wave, but also behaves as a particle, photons. And the amount of energy in the radiation is inversely proportional to its wavelength. Inversely proportional to its wavelength. And so you can see right here, smaller wavelengths have tons and tons of energy. I mean, these things obviously like X-rays, cellular damage can happen, albeit it's rare and your cells are really good at preparing and stuff. Microwaves, radio waves, no possible cellular damage, way too long. Okay, UV light can damage, and then of course gamma, which is those like nuclear waves, can definitely damage. And then here's Roy G. Biv. There's the visible light spectrum, and all the way from 700 to 400, okay? So photons can be absorbed by specific receptors, okay? So a photon is absorbed by some sort of molecules. We're just kind of getting background information here. And then we can raise that molecule to an excited state, okay? And therefore it has, you can see on this side, it has more energy up here, okay? And so pigments are able to do this in plants. There's a crazy amount of physics here, and hopefully this points you to like complexity and the argument that there is a creator. But pigments absorb wavelengths in the visible light spectrum. So chlorophyll, here we go, so let's dig in. Chlorophyll absorbs blue and red really well, but the remaining light is reflected, okay? And so an absorption spectrum is a plot of light energy absorbed, and then an action spectrum is the plot of biological activity against the wavelength. So let me show you an example of that here in a second, okay? So an absorption spectrum is a graph that plots a pigment's light absorption versus wavelength. So chlorophyll A, there's different chlorophylls, so chlorophyll A suggests violet blue and red and light works best. Accessory pigments, so these are kind of like other pigments that help the plant absorb sunlight. Chlorophyll B and other pigments called carotenoids. These guys help photosynthesis happen, okay? And so you can see right here, so you can see right here that which wavelengths of light are most effective in driving photosynthesis. Well, you can see right here, absorption, we're absorbing tons of energy here and also tons of energy here, okay? And then here's an action spectrum. So this is a graph that plots the rate of photosynthesis versus wavelength. And so this is biological activity, and so you can see our activity is very low in like the green and yellowish area, and that is what's being reflected, okay? And that's just them on top of one another. So you can see the absorption spectrum of chlorophyll A and anochorous, and then the action spectrum, you can see right here that the biological activity increases when there's more energy being absorbed and decreases when there's less energy being absorbed. Okay, here's what chlorophyll looks like. Complex molecule, right? And so, I don't know about you, but I love fall. Florida, we don't really get fall, but I would love to, one day eventually go back and visit the fall leaves. I used to go every fall, and the fall leaves are beautiful up north. And so, but why does this happen? Well, it happens because plants are shutting down chlorophyll production because they are responding to the stimulus they're receiving, shorter days, colder temperatures. So they're shutting down stuff ready to go into winter. And so all these different colors are different pigments coming to the forefront. The pigments are arranged. So a photosystem is that type of pigment, okay? And so here's what a photosystem looks like. So you can kind of see, and this is, again, just a brief taste of the complexity that involves photosynthesis. But you have a light harvesting complex. You have these different chlorophyll molecules that are absorbing light. And then you have kind of like this reaction center chlorophyll molecule. And all of that filters in, we're absorbing light, and it kind of filters in this central place, okay? And then that central place is where we're going to have our primary electron acceptor. And that's where we're going to energize an electron. Again, we're greatly oversimplifying this, but you can see right here, look how complex this protein is. Just amazing kind of thing about. And so when chlorophyll absorbs light, it enters an excited state and then rapidly returns to a ground state, releasing an excited electron. So chlorophyll gives the excited electron to an acceptor. That the acceptor is reduced because it's receiving an electron and becomes oxidized. The acceptor molecule is reduced. There that is. Sorry for saying it twice. So the reaction center, this is how, again, very briefly, this is how we've converted light energy into chemical energy, which is the foundation. This is the foundation of life. And this is kind of in a picture here, okay? So absorbing light, photons. We're kind of like transferring all the light right here. And then we energize an electron, go back to the ground state, and that electron is transferred to a molecule that accepts it, okay? And where does it accept? Well, the acceptor is NADP+. We take hydrogens and electrons, and we reduce NADP+ into NADPH. This is going to be a huge molecule as we go on in photosynthesis. And so ATP is produced chemiosmotically during electron transport. There is a little ATP produced in the first part of photosynthesis, okay? And so the light reactions consist of two alternate electron pathways. Okay, my bad, sorry. The non-cyclic pathway and the cyclic pathway. They both capture light energy with photosystems, okay? And so we're kind of like absorbing solar energy. Where is it happening? Thylakoid membranes. Both pathways produce a little ATP, and the non-cyclic pathway also produces NADPH. Okay? And so here's a picture here. So here you can see this is the non-cyclic pathway. Two different photosystems. Photosystem 2. Yes, 2 comes before 1. They found 1 first. It's okay. P680, so it absorbs light maximally at 680 nanometers. Photosystem 1 absorbs light roughly at 700 nanometers. So you absorb light, you have this energized electron, we transfer that down, it's going down because it's losing, potential energy. And so going down, electrons, going through these proteins, and so ATP is produced a little bit, right? And then we energize the electron again at photosystem 1, and we transfer it to a carrier, electron carrier, and that is NADPH, which is the reduced form of NADP+. Okay? You might be wondering, where do these electrons come from? Where do they come from? Well, they come from water. Okay? Water is split or broken down into hydrogen, oxygen, and electrons. And there's our electrons that are funneling into photosystem 2. Okay, here's just another picture of that. And then this is just a simplified version of like, okay, we're going to energize these electrons using light. Going to make a little bit of ATP at the mill. Going to energize them again, and we're going to store that energy into NADPH. And that NADPH is going to the Calvin cycle. And so talked about this already, photosystem 2 and photosystem 1. The electron is passed through a series of thylakoid membrane-bound carriers to a final acceptor, and so a proton gradient is generated and used by ATP synthase. We've heard that term before, the enzyme, right, ASE, to make ATP. Okay, the non-cyclic pathway, which is what we've been talking about, takes place. Okay, so where does it take place? You know where, what happens? It takes place in the thylakoid membranes. PS2 and PS1. PS2 captures light energy, causing an electron to be ejected from the reaction center. Electron travels down the ETC to PS1. The electron comes from water. This causes hydrogens to accumulate in the thylakoid chambers, and proteins in the ETC pump actively, okay? Using the energy found in the electron, okay? Where's this energy found to pump something? In the energized electron. And the hydrogen ion gradient is used to produce ATP, right? Because it's going downhill through the enzyme ATP synthase. PS1 captures light energy and ejects an electron. The electron is transferred permanently to a molecule of NADP+, causing NADP production. So ATP synthase is that enzyme, oh my bad, not used to using Keynote, is the enzyme that makes ATP. Okay, so the flow of hydrogen back across energized ATP synthase, and that's called chemiosmosis, when we're allowing hydrogen ions to flow downhill producing ATP. So we just talked about the first reaction, the light reactions. Okay, there's two reactions, and this is a nice picture here, but there's two reactions in photosynthesis: the light reaction, or the light-dependent reactions, and the Calvin cycle, and this is also called the independent reactions. Okay, and so here's a great picture. We're going to color something here in a second in notability, but here's a great picture here. So can I zoom in? I can, I guess. Okay. Here's PS2. Electrons are coming from water. Again, need to be able to flow with this. We're pumping hydrogens across in the thylakoid space. Okay. And those hydrogens eventually will flow through ATP synthase and chemiosmosis, the diffusion of hydrogen from the thylakoid space back down to the stroma. Powers ATP synthase. It looks like a motor. It turns. I'll show you some videos in class, hopefully, and it's pretty impressive. But the electrons are going down the gradient and then to PS1, and then we store it in NADPH. Okay, wow, that's really tiny. Okay. So this is where I would like to go to notability. Let me see if I can do this now. Cool. All right, let's scroll all the way down. Sorry. I'm sure there was an easier way and smoother way to do this, but nonetheless. And so this is when I want you to take out your color pencils or some sort of coloring and device, and I'm going to walk you through the light-dependent reactions, okay? It's very important that you understand this. Very important that you can do what I do right now on your own. And so let's just start with blue, okay? So I'm going to start with blue. And because blue is water, right? Well, why is blue important? Because blue is where we get our electrons from, okay? And I know it's not like the best coloring, but maybe I can make it smaller. There we go. Blue is where oxygen gas is produced, and we release some hydrogen ions from the splitting of water, photolysis, okay? Those, that electron is energized at PS2, so this is the traveling electron, and it goes to PS1, and then PS1 energizes it. It goes up here, and eventually we give that electron to NADP plus a hydrogen ion, and we produce NADPH, and that goes to the Calvin cycle, okay? So that's where the electron kind of goes from. Everything, of course, can't do anything without sunlight. And so sunlight is necessary, and sunlight is hitting simultaneously photosystem 2 and 1 at the same time. Okay, but just for ease of learning, we're going to just talk about photosystem 2 first. So photosystem 2 is right here, okay? And we are taking light and capturing that light via pigments, chlorophyll. The main one is chlorophyll A, although there's accessory pigments, chlorophyll B, and other types of pigments. And then here's photosystem 1 over here. And you can kind of see that this is photosystem 1, energizing the electron as well, okay? Now, what do we do with that energy in the electron? Well, I'll make this, red, I guess. but we are pumping hydrogens across this membrane. Look, so hydrogens are going this way, okay? And so we're pumping hydrogens against their concentration gradient, hence the word pump, right? And so those hydrogens are flowing then through this molecule, and this molecule right here is ATP synthase. Okay, this is just a diagram. And so ATP synthase is the molecule that has, the ability to let hydrogen ions flow downhill. And as they flow downhill, it turns, literally this enzyme reacts and it turns. I'll show you a video in class. And it makes ATP from ADP, which means there's two phosphates, plus inorganic phosphate. And so ATP also goes to the Calvin cycle, okay? And so that's the purpose of creating that hydrogen ion, concentration gradient, those protons. And so this is called oh man, pen's not working. This is called chemiosmosis, okay? And then, yeah, these proteins in here, you don't have to know these, guys. I don't know what color I haven't used yet, black. Yeah, you don't need to know all these fancy names of these proteins. That's beyond the scope of this course. But there's, of course, this is the electron transport chain. So there's a lot of different proteins involved here, right? This stuff just doesn't magically happen. We're talking enzymes that are created via the central dogma, and everything like that. And it's so complex, right? We're just scratching the surface of this stuff. But that's the electron transport chain. That's the ETC, okay? And so being able to understand, okay, the last thing I want you to understand is where all this happens, okay? So the where is important. The stroma is the fluid inside of chloroplast. First off, we're in a chloroplast, and specifically in a thylakoid membrane. Okay, so here's the thylakoid membrane. Here's the thylakoid interior space, okay? And so you can see that the hydrogen ions are pumped into the space, into the interior, and it flows out. And then ATP and NADPH are in the stroma, and they're going to go to the Calvin cycle, which is just, again, more enzymes floating around in the stroma. So again, we're in the stroma here, okay? And so, yeah, I hope that's helpful in understanding the non-cyclic pathway of photosynthesis. This is the light-dependent reaction, okay? And so ATP is needed for carbon fixation pathways. We got to build, right? We're building stuff. And so we have to be able to build. Cyclic electron transport uses only photosystem 1 and produces only ATP. 1 year, I don't know if it was 2 years ago or something, but they actually put this on the exam. And I covered it probably for 30 seconds in class. That was pretty impressive, which probably means it's not going to be on this year's exam because it was so recent. But nonetheless, let's dive into it together. And so here's cyclic electron transport. Okay, so here we go. PS1, reaction center. Okay, energized. Boom. There we go. Here's all the electron carriers and proteins. They're all reduced. We're making some ATP, and the energy from the electron flow is captured for chemiosmotic synthesis of ATP. Right. So whenever you see chemiosmosis, you should be thinking hydrogen ion, like a dam. Right. The dam, a lot on one side of the membrane, not much on the other, flows downhill. And so the last reduced electron carrier passes electrons to electron-deficient chlorophyll, allowing the reactions to start again. So the electron is just kind of cycled. Hopefully you see that. It's kind of cycled around. So that's why it's called cyclic, okay? And so here's the questions for this lesson. What color of light is least effective in driving photosynthesis? Explain. And the light reactions, what is the initial electron doing? Where does the electron come from? And at the end of light reactions, where are the electrons? Okay, we're talking about the light-dependent reactions here. And then a great, last one, a little bit more challenging. In an experiment, isolated chloroplasts placed in an illuminated solution with appropriate chemicals can carry out ATP synthesis. Appropriate chemicals, okay. Predict what will happen to the rate of synthesis if a compound, predict, that's your action verb. If a compound is added to the solution that makes membranes freely permeable to hydrogen ions. Okay, so that's a great question, because you got to take the concept and something new is happening, and then you have to relate hopefully what you've learned and what you've been taking notes on, and think about, go back and review. But what's going to happen if a chemical is added, a compound that makes membranes freely permeable to hydrogen ions? Hope this was helpful, guys. I'm really proud of you.